can we move the needle here on morbidity, mortality? How do we really make this a very good story? What I'm going to argue for in the next few minutes is that we've got to completely transform what we've been doing, and even transform the way we think about the problem. So I'm going to talk about three, we'll call them disruptive innovations. I'm not sure that's the best term, but I've been using the same formulation for several years. It's not actually that innovative, it's not that disruptive anymore, but it was when we started a few years ago. And I thought I'd just share it with you and just unpack it a little bit so you have a sense of where we're coming from. The first and maybe the most contentious is to be able to say that these are ultimately brain disorders. And that's not to say that we're not interested in the mind, we're not interested in behavior. It's only to say that there is a biological organ underpinning both normal and abnormal behavior. And what we have to get to is a point where we understand that at the level of at least the circuitry. It's rather pathetic for me that this is a field that has seen the brain basically as a bag of soup, in which when you're low in serotonin, you have depression, and maybe you have a little too much dopamine on board, you have schizophrenia. And that all came about because of medications that we thought were useful, and trying to understand what the medications did, which would be like saying that whenever you have a headache, you have 2 little aspirin. The key here for us is to begin to understand, as some people have said, we have to go from soup to sparks. We have to begin to think about what is the circuitry here, and how do we get to a point where we can understand the underlying code that may be essential for mood regulation, for executive function, for impulse control, all of that. These do result from complex genetic risk, which shouldn't be too surprising to anybody. What that really means is that it's a combination of experience, working on the genome, and through epigenomics, changing the way that genes get expressed, the way the genome gets read, as well as family history, which drives a very complex story, and we'll say a little bit more about that. And finally, those two together, the fact that these are brain disorders and that they're partly coded by genetics and epigenetics, means that we can think about these as developmental disorders. I already said that this is the group of illnesses that start early. It's almost what defines mental illness is the fact that these are almost all neurodevelopmental disorders. And so we're talking about how early experience and genes work together to change the way that circuits get laid down and what that means. Well, as I said, this has been basically the formula that I have been proposing for about 6 or 7 or 8 years. I'm now beginning to forget. But the reason I'm telling you this now is, A, we're still very much on this track. In fact, it's very much the NIMH strategic plan. So if you want to know where's the money, which is usually what people want to know when they come to hear me talk, this is partly the answer to that question. What is worth telling you is that the answers are also changing in a lot of really interesting ways because the tools to do this kind of work are changing. And I'd like to just zip through each of these very quickly, unpack them one at a time. I'll use those little icons to the right to guide you around how things are changing. We can start with a video which was released about maybe a year ago, but less than a year ago. This to actually start. This is a method called Clarity, and was developed by Carl Geiseroff, just down the coast at Stanford. And what you're seeing here is a 5-1 express brain, that's a mouse brain in this case, that has a hydrogel infused in it. All of the lipids are taken out of the brain. This is post-mortem material. And when you take the lipids out, you put the hydrogel in, it holds the brain's integrity and allows you to glide through. You have to put your seatbelt on here. If you get seasick or sick, I apologize. But it's a chance for the first time to be able to have what we call 3-dimensional neuroanatomy, to be able to see the relationship of cells throughout, in this case, the mouse brain, so that you can trying to begin to actually see the circuitry in a way that's entirely preserved. There are two pieces of this that make it even more amazing than the kinds of neuroanatomy we've done in the past. One is that you can do this in human brain as well as mouse brain. You just need to have a postmortem specimen that allows you to soak out the lipids and put in the hydrogel. The second, which is probably more critical, is that this is, as I mentioned, this is Thy1 with a GFP state, so you can see just those cells that express Thy1. But you can then begin to infuse this hydrogel with antibodies and DNA and all kinds of things that you're interested in to stain for all sorts of things, repeatedly. Stain, wash, stain, wash. And the result is you can begin to see the molecular neuroanatomy of the brain in 3 dimensions in a way that we just haven't conceptualized before. And that really helps us to see the relationships if what we're about is trying to understand circuits. This is way beyond soup. This is really getting us to the wiring of at least the mouse brain and now the human brain as well. Of course, the downside here is that this is post-mortem, so you're not going to be able to do this very often, and it's not going to be very helpful in making a diagnosis, or if it does, it's way too late. What you're looking for is something that will allow you to be able to understand how the brain works in real time. And so there's another story to tell, also from Stanford. This is Carl's colleague, Mark Schnitzer. By the way, I should mention, Carl's a psychiatrist, as many of you probably know, as well as an engineer. Mark is in, I think, the Department of Bioengineering, and he's created these interesting mini microscopes, or micro microscopes. He calls them micro endoscopes, but that's a much less interesting name. What they are just very, very tiny devices that you can put on the head of a mouse or an experimental animal, and they have an objective that goes right into the brain, and then you can begin to look in real time for weeks at a time, if you want, at an underlying field to see what's happening in terms of not just a few cells, but thousands of cells and how they're working. In this case, I think he's looking at about 1,000 or 1,200 hippocampal cells in an area that's important for place. So these are place cells in the hippocampus. And what he's doing, and these are, in this case, these are cells that have been engineered to express GCAMP3, which lights up when the cells fire, when they're activated. It's a calcium indicator. And what you'll see here on the right is, the pattern of firing when an animal, a mouse in this case, is running a maze. But not for very long. I should probably not move my pointer. Okay, let's try this again. Okay, so you see the firing rate of the cells on the right hand. The left hand is just where what the cells look like when they are, and when the animal is still. And you can see that as this animal runs around, there's an interesting pattern of firing. And the question is simply, can we get that code? Can we figure out how in this part of the brain that's important for spatial orientation, how is the information being encoded? And what is the code? And Mark and his colleagues have been working on this, and they're not quite there. I'd say they've cracked it partly. They've cracked it well enough to know that just by looking at the pattern of firing in the right-hand panel, they can predict which arm the mouse is in to within 1 mouse point. Pretty good, but they're not quite there. But it tells you, again, that in the world of trying to understand disorders of circuitry, we're making progress, and we've got tools that are really going to help us to get there in the near future, especially for mice. The bigger problem, of course, is what we can do with the human brain, but that too is getting really interesting. This is from the Human Connectome Project. It's a very large project supported by the Neuroscience Blueprint at NIH, in which groups between MGH and another project which is going on at WashU and the University of Minnesota is essentially trying to get the wiring diagram of the human brain, much the way that the genome got the DNA blueprint of the human genome. This effort, which is about now maybe 3 years old, is going to be drawing to its first endpoint in about a year. And at that point, we'll have very high resolution connectome images, both functional and structural, from 1,200 people, 300 identical twins, all of which will include a lot of phenotypic data with cognitive testing, genetics, a whole range of different kinds of information, and entirely open to anybody who wants to interrogate it. So this is creating this spectacular database. If I were a student coming into this field, whether it was in San Francisco or Singapore, I probably wouldn't even start doing scans. I would just start by asking really fundamental questions that you've got from all the data that's being put up there, which no one has even begun to interrogate yet. I think there are now about 150 of the scans that are already registered on the Human Connectome site. And it comes with analytic software at the same time. We're in a whole new room. For those of you who don't do this kind of science, it's just so incredible to me that the discoveries from this project are going to come probably more from Beijing and Bangalore than Boston, because all of this is public, and anybody anywhere with a good idea who likes to do this kind of stuff can begin to interrogate it and give us information. But right now, it's just data. You know, really exciting couple of years. By, as I said, I think by next June, we're looking at having about 1,200 of these images available. We haven't yet gotten to the point where I think we can say with certainty that we know what the circuitry is for any of our disorders, but we're getting a lot closer. We've got sort of the first primitive maps of what areas would be involved, and you can say as much as to say that these are not completely overlapping or identical maps for the different syndromes that we think about, but there's an enormous amount of individual variation. We're not at a point where any of this would serve as a biomarker, but just as kind of a feeling for what it might look like in the future. This may be the 2023 map of what having an affective illness could look like, where mood dysregulation involves many, many different parts of the brain and several kind of interwoven and overlapping circuits. This is from Helen Mayberg's work, and as Helen has pointed out, already one way to think about this that may be helpful is to realize that different interventions affect different parts of the circuitry. So medications working in one area, psychotherapy in another, and what she does, deep brain stimulation, going after this area 25, hitting one of the many governors. Conceptually, I think what's important here, as we go from soup to sparks, is not to think about there being a site in the brain for depression or for mood. It's just not the way the brain works. There are categories in the brain, but the brain is a very circuit-based system with lots of overlapping circuits that have lots of redundant activity. We don't fully understand exactly what this emergent property that we call mind and mental behavior, but we do know enough to know that almost every area is connected recursively to thousands or hundreds of other areas. And so the ultimate maps are going to be this kind of very complex picture. And this is still kind of a macro map that will probably have to get much more resolved at close to a cellular level to begin to really be informative. So that's a quick rundown on thinking about brain disorders as circuit disorders. When we talk about them being genetic, what does that mean? Well, it means essentially that there is, like everything else in the body, a genetics that underlies how the brain gets organized. And the piece of this that you need to think about is the enormous variability that occurs in the genome, and the way in which that variability combined with very early experience, especially much of it prenatal and some of it postnatal, leads to the laying down of pathways and laying down to the very circuits that we were just talking about. The point I want to make here is that none of this is going to specify for what we call schizophrenia or what we call depression or bipolar illness. it just doesn't appear from what anybody can determine that our genomes ever had a chance to read the DSM in any of its variants. What it does appear is that the genome variation does relate in very interesting ways to specific circuits that we know about, because that's all played out at the level of cell biology and how cells wire and fire together. And that those circuits, in turn, don't specify for an illness, but they specify for specific cognitive domains. And this may be hard to read in the back, but this is episodic memory, and working memory, and emotional regulation, and reward. And those are 4 of about 100 that you can come up with that are the real workhorses of what the brain does. And what we need to be thinking about as we think about disorders is how those cognitive domains play out in terms of our disorders. More about that in just a few minutes. But if we just go back to this level, at the level of genetics, over the last year, this has just gotten so much more complicated. It's really interesting in ways that I find a little disruptive and actually disturbing. ENCODE is a project that's supported by the NIH to give us very deep information about the structure and the sequence of the human genome and all of the things connected to it. And I tend to separate the history of modern science into DE and AE, before ENCODE and after ENCODE. Before ENCODE, we thought about most of the genome as kind of junk. We realized that about 1.6% of it was important for genes that encode proteins, but the rest of it was, we didn't call it garbage, because that's what you throw out. We called it junk because it was still there. Evolution held on to it, not knowing what to do with it, but it was still around. It's not junk anymore. What we now know from ENCODE is that 80%, not 2%, is transcribed. We've been looking at these big protein coding things that we call genes, but those are probably just one little part of what the genome does. The genome has something like 20,000 different RNA sequences that don't code for proteins, but probably code for regulatory, interesting kinds of protein, or interesting kinds of messages, and lots of protein binding sites, lots of transcriptional regulators that we didn't even know about. And we've now begun to rethink the genome, not as code, essentially, but as something that has lots of different kinds of information embedded